Today we are pleased to have Priya Moorjani come to talk to us from Berkeley. Priya did undergrad in computer engineering at the University of Mumbai and a master's in bioinformatics at George Washington. Then did a PhD in genetics with David Reich and Nick Patterson, postdoc with Molly Prozorski, and has been at Berkeley since 2018. And she's done a whole bunch of really neat work. She's kind of a wizard at using genomic variation data to learn about ancient history, especially neat stuff using recombination, information from recombination to date things. And she's been doing work on disease mapping as well, and work on mutation rate variation, which I'm particularly excited about, and I think was what we get to hear about today. So welcome.
Very much. Thanks, Peter, for that kind introduction. And thank you, everyone, for having me virtually. It's wonderful to connect and keep science going even during these times. Today, I'm going to share some unpublished work from our lab. And so I particularly love to hear feedback from you. This is a project looking at variation in mutation rate across human populations. And just to mention, I'm happy to take questions in between, so feel free to just unmute and ask a question, or if you prefer, I can also take questions at the end, of course. I can try to keep track of the chat, but it's often harder to sort of look at the chat while talking. But let's talk about germline mutations. Germline mutations are the ultimate source of genetic variation, and they are the fuel for evolution. They are the cause of many heritable diseases. And because mutations occur steadily over time, they provide a record of the time elapsed. And so for us in particular in our lab, this is a really neat feature of mutations that we try to leverage in building molecular clocks for learning about the timing of different events. And the question of mutations and the earliest estimates of mutation rate are actually, they date back to 1930s when it was not even appreciated that DNA is the heritable material. The earliest estimates are from Haldane, who compared the rates of hemophilia across different families to try to infer the rate of mutations. In recent years, because spurred by how cheap sequencing has become, there are now whole genome sequences across different species, across individuals within a population, and also of many pedigrees. And this has really changed fundamentally how we think about mutations and what are some of the factors now we need to think about when we think of mutagenesis. And for me, this question really became forefront with this one finding where in humans, when people estimated mutation rates, they saw major discrepancies in mutation estimates across pedigrees and phylogenetics. So in phylogenetic data, when you estimate mutation rate, you compare humans to, say, another species, and given an estimate of the time based on fossil record, you can then infer a mutation rate. And this provides an estimate that reflects an average rate over millions of years. But with pedigree sequencing, we can now just sequence the genome of the parents and the offspring and just learn directly count the number of mutations that happen in one generation. And this provides an estimate that reflects the current estimate of mutation rate. And when you compare these two estimates, what you find is that they differ by almost twofold. And this really has a major consequence in our understanding of the timing of different evolutionary events. For instance, if you consider the human-chimpanzee divergence time, that's about 6 million years if you use the faster mutation rate, and that's about 12 million years if you consider the pedigree rate. And same with the human old world monkey divergence time, they almost scale by twofold, such that the human old world monkey divergence time is about 6 million years. And in particular, these later estimates are very inconsistent with the fossil record because there are many fossils at this time scale, and those suggest a much earlier time of split of these species, whereas the pedigree estimates suggest a much older time. Now, us and others have written a lot about this, and there are and looked at it in a number of ways, and there are many technical factors that could possibly impact this effect. So, but possibly due to selection, there could be effects on the phylogenetic estimates, or due to technical factors, there could be effects on the pedigree rate. But what many have also suggested that maybe the biological reality is that possibly mutation rates are evolving over time. And there could be many factors that could be impacting the evolution of mutation rates. And so this is what I really focused my postdoc on, and this is the work that we are looking into now in my lab also. Now, from pedigree studies, we can now precisely infer the mutation rate in one generation and actually start looking at different factors that can impact this rate. The way to do this is to sequence the parents and the offspring and then just count the number of de novo mutations that occur in one generation. This is a figure from Decode Genetics where they have generated now data for 2,500 pedigrees, and you can start very precisely estimating how the age of the parents at conception impacts the mutation rate. So you can phase the mutations, so assign them to either the paternal or the maternal genome, and then study how the age affects the rates. And by doing this, we find that for every year increase in the father's age, the offspring gets about 1.5 additional mutations. We know that this is because spermatogenesis continues post-puberty in males. And for mother's age, we find that the effect is lower, but there's still a significant effect of the maternal age. And we find that for every year increase in the mother's age, the offspring gets about 0.4 additional mutations. While oogenesis stops at the birth of the mother, these mutations could still be accumulating due to DNA damage and other factors. And that is consistent with the patterns that you observe in the de novo data. We can also look at not just the overall number of mutations, but also how different types of mutations depend upon the parental age. So considering, say, C2a versus C2t mutations, also accounting for sometimes the context in which the mutation occurs, so considering the neighboring base pairs. And we can see that even different types of mutations have distinct dependencies of parental age, and some mutations like C2G increase with the mother's age faster than the father's age, while other mutations increase with the father's age, mutations like T2C or T2G mutations. And this means that not just the overall mutation rate, but we actually need to decouple the mutation rate into a mutation spectrum. By spectrum, I mean proportions of different types of mutations that an individual inherits. So these are sometimes represented as a bar graph where you have different types of mutations. The y-axis shows you the proportion, or you can consider it as a rainbow plot where the x-axis is the proportion and the length of each of these colored segments is the proportions of a particular type. And in my talk, I'll use many of these different representations because each study has chosen to represent it their own way. And so just to avoid confusion, I'm showing this earlier. So we first looked at how the mutation rate differs across different primates. So how much variation is there in closely related primates? As you know that there is almost the base composition across primates is very, very similar. And so chimpanzees and humans have 99% similar genomes. And so if we compare these species, we find that actually for different types of mutations, there are still significant differences across different species. If we compare apes versus old world monkeys, the overall substitution rates differ by about 40%. And if we compare apes and New World monkeys, the substitution rates differ by almost 65%. And even within Apes, if we compare human and chimpanzees or humans and orangutans, there are up to 10% differences. And in these analyses, we have tried to control for selection by removing conserved regions, by looking at these analyses just in ancestral repeats, which should be to first approximate neutrally evolving. And these results are similar, and others have published similar estimates also. And as I mentioned, not only the rates, but if we also break down these different types, we find that there are significant differences across different species. And more recently, it's also been suggested that across human populations, also the mutation rate differs. Now, in humans, all the replication machinery is the same. And yet, when you break mutations down by the type, you find that across Europeans and Africans, mutation rates differ across different groups. One, the most significant among these signals is the signal of TCC to TTC, which appears to be present at a higher rate in Europeans compared to other groups. And if you stratify based on the frequency of this mutation, you find that this mutation increases as a transient pulse in Europeans, and then at present, it has decreased back where it's similar across human populations. Even within a population, we can ask how mutation rate changes over time. And to do this, what we first did is we just divided the mutation, we just divided mutations by the frequency that they occur. So if you just consider singletons and then estimate the proportions of different types of mutations, you get the mutation spectrum just in that one population. You can then repeat this exercise for, say, doubletons, common variants, and just see how the mutation rate is shifting over time. Now, as you know, these, while we can see that the mutation spectrum, even in one single population, changes over time, this is impacted by many different factors. In particular, demography could have a big impact on these patterns. So from a pedigree, at a pedigree end, the mutation spectrum mainly depends upon parental age. We find that also the DNA damage and repair processes in the parental genomes can impact the mutation spectrum. Possibly environmental exposures can impact mutation spectrum. And over time, the mutation spectrum is further impacted by other factors, demography and selection, but also biased gene conversion. And biased gene conversion is the process where, when in a heteroduplex you have you have inconsistency, there is a bias towards repairing the inconsistency, the mismatch towards certain types of bases, towards the C to G bases. And this works like selection, and over time there is bias towards becoming a more GC genome. But there are other factors that counteract this, especially methylation, which pushes the genome back towards A to T. And so this maintains a balance. And given that we know that CPG mutation rates are tenfold higher than other types, this bias is a non-significant effect also. And so a question we asked is, what are some of the factors that are impacting the mutation differences across human populations? For this work, we collaborated with Ziwei Gao, who is a professor at University of Pennsylvania, and Yulin Zhang, who's a PhD student in my group. And there are three main questions we asked. How many independent changes have occurred in the mutation spectrum in humans? When and in which populations these occurred? And then finally, what are some of the causes of these? In particular, how does generation time or the mean age of reproduction impact these factors? Now, to understand differences between human populations, we developed a framework where we first estimate the mutation age of each mutation. So we take genetic variation data from human populations, and using methods such as RELATE, which are coalescent-based methods, we try to infer the age of each mutation. We also then try to control for biased gene conversion. So rather than comparing the mutation spectrum across different populations, in order to control for gene conversion, what we do is we compare pairs of different mutations. So we compare pairs of mutations that are matched for effects of biased gene conversion. So comparing T2C to T2G mutations, or that are both favored by biased gene conversion, or comparing C to T to C2a mutations that are both disfavored by biased gene conversion. And then we also account for effects of methylation by looking at CPGs and non-CPGs separately. So rather than comparing the mutation spectrum in one population or across populations, what we do is we compare these polymorphism ratios. So just to walk you through what our results look like, this is the same plot as the one on the left, but now we are comparing ratios of two types of mutations. So each panel is a ratio of two types of mutations. On the y-axis is the age of different mutations. So first we take the variation data, we estimate the age of each mutations. We bin these mutations in different bins. So say 0 based on when they occur. So a bin of say 0 to 48 generations, a bin of 48 to 107 generations. And then in each of these bins, we take all the mutations and estimate what is the proportion of T to C versus T to G. We perform some bootstrap resampling to try to estimate the standard errors, and then repeat the exercise for the other mutation age bins and also for other mutation types. And when we do this, we can then also compare subsequent bins, and in doing so, in Europeans, what we find is that there is one signal that is significantly changing over time. And this signal almost leads to a 15% change at non-CPG to CPG C to T to C to A mutations. And this signal is the same one that has been previously identified by Harris and Pritchard as the TCC to TTC signal. And so we were really happy to replicate their findings, and also that even after controlling for effects of biased gene conversion, we still see this effect. So suggesting that it's not being driven by biased gene conversion alone. We can then repeat this exercise for multiple populations. Now here, each point is a different population. We look at Europeans, East Asians, and Africans. And then in each bin, we can now compare how do the three populations differ from each other. And we can see over time how are these differences occurring. Again, we find, in this case, we find three main signals. The first one is the TCC to TTC signal that we found in Europeans, and also what Harris and Pritchard have previously documented. We also find another signal. The signal occurs at differentiating C to G to T2a ratios across all three populations that we considered, so across East Asians, Europeans, and Africans. And then we found a third signal which differentiates Africans and non-Africans. And this signal is present at much older ages, in fact, predates out of Africa migration. So now let's look at each of these signals in turn. The first signal is the European specific acceleration of this C to T mutations. We previous analysis looking at the mutation signatures from cancer genetic literature has suggested that the signal or the signature is enriched in UV exposed due to UV exposure. Matheson and Reich, and also I think Harris and Pritchard also showed that beyond this one type, the TCC to TTC, there are other mutational signatures that also potentially are differentiated across human populations. So what we first did is we looked at how is our signal enriched if we just consider the specific signatures that they have identified. In doing so, we find that the signal is present. If anything, it's even more significant than when we look at all sites of this type. And now when we could also do the converse where we remove these signatures and then compare human populations. Interestingly, even after we remove the signal, we find that there are still differences across human populations. And this suggests that the TCC signal may not only be driven by UV exposure, but possibly there could be other factors that are also impacting these changes. It could be occurring due to generation time differences across human populations, as Marcia and Scav have recently suggested, or due to genetic modifiers that are segregating in human populations, as Harris and Pritchard have suggested. Next, we look at the variation in the C2G to T2a mutations. When we saw the signal, we were actually really excited because as you remember from an earlier slide, I highlighted that C2G mutations are disproportionately increasing with maternal age. So we were quite convinced that this signal is driven by maternal age differences across human populations. The signal is really strong and almost explains majority of the maternal age effect. And in fact, from the earlier study by Decode Genetics, they also identified that there are certain regions in the genome where these mutations are concentrated, and there is almost a 50-fold increase in these mutations in these regions. They call these mutations as maternal hotspots. And almost 57% of the maternal mutations fall in these 2 or 3 regions across the genome. And so the first thing we did is we looked at our signal and we tried to exclude these maternal hotspots to see if the signal goes away. Interestingly, we actually found that we were wrong and the signal is still present even after you exclude the maternal hotspots. And in fact, if you focus on the maternal hotspots, you no longer see any significant differences. And so while you can have your hypothesis, data can always tell you if you're right or wrong. And so next, what we did is we tried to compare this ratio with by changing the denominator. The reason to consider ratios was to control for biased gene conversion. So if we consider other denominators, we might not be controlling for biased gene conversion. But given that we know it's not driven by biased gene conversion, we can still leverage if the numerator or the denominator is driving the signal. So first we compared the T2G. First, we changed the numerator by replacing the numerator with T2G instead of C2G. We still see the signal. We replace the numerator. We replace the C2G by T2C. We still see that the signal is present. We can now also just replace the denominator where we replace C2G, we replace the T2a by C2a so that to check if the signal is driven by C2G. And in this case, we find that there is no difference. So this signal is likely being driven by the T2a mutations. It seems to be that these mutations are present at the highest rate in East Asians, and then they decrease between Europeans and Africans, but actually even Europeans and Africans have slightly different rates, suggesting that this is not necessarily a East Asian specific signal, but may also be some transient modifier or other factor that's segregating in all 3 populations. Next, if we look at this T2C to T2G signal, this is the most interesting signal because at this point, at such an old time in evolution, all 3 human populations should be one population. So we should not expect any differences across Africans and non-Africans, because out of Africa migration has been dated around 2,000 to 3,000 generations. And so looking at very old time scales in human evolution should not suggest any differences across human populations. And yet we saw that there were significant differences. When we first saw the signal, we thought this was primarily driven by technical artifacts, possibly a subset of ancestral alleles that were being miscalled as derived alleles. So we did a lot of work to try to rule out technical issues. I have a backup slide that I can show you if you're interested. but we redid our analysis by remapping to chimpanzees rather than using the human ancestral allele. We looked at regions where of just European ancestry or just African ancestry. As you might know, the human reference genome has mosaic ancestry, but previous work in PopGen has actually characterized ancestry. So we could focus on just looking at the European chunks of the human reference genome or just the African chunks. And the signal is still present even after considering all these technical artifacts. We then thought of what might be other factors that differentiate Africans and non-Africans. And obviously, most of you might know the history of Neanderthal gene flow could possibly have an effect on this. And so we looked at regions of Neanderthal ancestry and recalculated these ratios. Interestingly, we found even after we remove all the regions of Neanderthal ancestry, we find there are still significant differences across human populations. Next, we looked at a recent publication where Durvasula and Shankararam have suggested that even Africans have some archaic ancestry. This is due to a gene flow that possibly occurred in the common ancestors of all modern humans, where there is archaic ancestry now, due to which there is about 2 to 20% archaic ancestry in all modern humans. If we now go in and remove the signal that they studied in their paper, we actually find that there are no significant differences across human populations. Now, what we are doing here is their signal is focused on looking at sites, looking at the site frequency spectrum conditional on archaic populations. So assuming, looking at sites where archaics have a derived allele, and then you look at how what are the frequencies in human populations. Here is the graph for Africans in blue. The little circles are related to Neanderthal ancestry, and the dark blue is Denisovan, and the dotted colored lines, which don't matter here, are just their model. They do a lot of simulations to show that the signal can be explained by archaic ancestry based on their simulation analysis. If we focus on these sites, we find that there is maximum difference across Africans and non-Africans, and there's almost up to 6% difference across these groups. And when we remove these signals from our analysis, we find that there are no differences across human populations. We again tried to understand if this is related to T2C or T2G mutations, again by comparing our ratios, by changing the denominator in this case, and we find that it's primarily driven by T2C mutations. So possibly the T2C mutations changed in this common ghost archaic ancestor, where for some reason, this group, due to possibly generation time differences or a segregating mutation modifier, they had a higher proportion of T2C mutations compared to, say, T2G or other types of mutations. And we also have recently started looking at what might be the mechanism behind this change. So we can look at these sites where there are differences across human populations, and then apply NMF or PCA-like methods, and then try to study what is the mutational signature that is driving this signal. This is work by Monica Arnila, a rotation student in my lab, and she found that the signal is mainly driven by three mutations occurring in three contexts. So when they are preceded by a C or G or A or G, or C or T, that's where the signal is primarily concentrated. If we remove this mutational signature, we actually find there are no differences across human populations, but we still don't know what the signature is related to. We are looking into the cancer literature and in cosmic signature literature to figure out what might be some of the factors that could be leading to an increase in T2C mutations. So now I want to get back to the first question that, another question that I asked is, if there are these differences, what is the role of generation time in leading to these differences? So far, I've talked about how we could use mutational signatures to look at other mechanisms, but now let's try to think of how generation time changes could just lead to these differences. And to do this, we leverage how we have two kinds of data, where in the pedigree data, we can study the mutation spectrum, but the pedigree data is really nice because it also gives, or you also have information about the age of the parents, and you can study how the mutation rate depends upon age and sex of the parents. We can then study the mutation spectrum in polymorphism data, which helps us understand how these ratios or spectrum changes over time. And so we could then learn about the dependencies of mutation types or ratios on age and sex, and then we can apply that to the polymorphism data to see if how the observed spectrum can be explained by age and sex. And so we took pedigree data from Decode Genetics. This is data for 2,500 individuals. A subset of this is what I showed you in an earlier slide. And we just estimated what is the ratio of, say, T2C to T2G at a particular age. You can estimate this at, say, age 20. You can repeat the exercise for different ages of the parents. So you can do that for age 40, and we find that the ratio decreases with age. We do something more sophisticated where we look at the regression model. We account for the uncertainty by performing bootstrapping of the data, and we estimate that this ratio decreases with age of the parents. Here I'm considering the mean age of the parents, so just averaging the age of both parents. We can also look at this ratio with age, as we have done before. For most of our comparisons for looking at generation time, we only compare the polymorphism ratio in Europeans, because it's possible that the mutational properties and parental age effects also differ across human populations. And so we just learn these dependencies from Decode Genetics data, which is primarily Europeans, and then look at the polymorphism spectrum in Europeans. And then we can simply compare what ratio does this observed ratio in polymorphism data, what generation time does it predict? And if we did that, we can then infer from this comparison, accounting for the standard error, that this would predict a generation time between 30 to 40 generations. 30 to 40 years, sorry. And we can do this analysis for each different time bin. And what we find with the T2C to T2G mutations is that they predict a relatively stable generation time after 30,000 years, but they actually, in most cases, predict a very old generation time between 30 to 40 years. We can then look at other ratios, looking at C2G to T2a. And in this case, we find that this ratio actually predicts very young generation times. And we can also do this analysis for, say, the C2T to C2a control for effects such as the TCC to TTC mutations by removing them both from the pedigree and polymorphism data. And so you can see this peak, which was present due to TCC to TTC. After removing these types, the peak decreases, but there is still a peak in the data. And we find that this ratio is actually not, many estimates at early ages that predate out of Africa are not consistent with the pedigree data. But we can put all of this together and estimate a generation time for every time window for each pair of mutation. And this is the summary of the results, where each panel is a type of mutation, T2C to T2G. For each of the bins, I'm showing you an estimate of the generation time that we infer using that ratio. And we can also estimate the uncertainty in this estimates. What really surprised us when we did this is that actually the patterns are not clearly driven by generation time, or generation time cannot necessarily explain the patterns we observe in the polymorphism data. So if you look at one time window, again, looking at the same 48 to 107 generation time window, what we find is that for some mutations, the generation time is predicted to be very old, while for other mutations, it's predicted to be very recent. And we can also control for these factors by removing some of the mutational signatures like TCC signature and mutation. While there's some shift in the generation time, the estimates are not consistent with other types of mutations. Even over time, we find that some mutations appear to be changing very slightly with age, while others appear to be changing non-monotonically with age, and others appear to be changing. In other cases, the patterns are very noisy. And this suggests to us that generation time cannot alone explain the patterns in polymorphisms. We have now done these analyses considering different models of parental age dependence, because one could argue that maybe maternal age has a slightly different impact compared to paternal age. And we have modeled those differences also by constraining ratios of maternal to paternal ratio to be 1.1 or 1.2. Because we might expect that paternal age is always longer than maternal age over evolution. But even in those cases, we cannot necessarily explain these patterns. So just to summarize, I showed you three different signals that we observe of changes across human populations. We see the first signal, which is the transient change in C2T mutations at non-CPG sites. We find a signal of divergence in the T2a mutations across three continental groups. And we also find a signal related to archaic ancestry that differentiates Africans and non-Africans. In brackets here, I have speculated what might be the possible causes that are driving these signals. The TCC signal the C2T signal could be driven by the TCC change or environmental exposure, or more likely genetic modifiers or generation time. The T2a signal, I think the pattern that it has seems unlikely to be explained by generation time. We know that it's not driven by maternal age effects, but it also seems quite unlikely to be driven by generation time, because it really differentiates all three populations, and the pattern is consistent over many generations. So it would almost be required that East Asians, Africans, and Europeans at one day decided that they would start having offspring at a particular age, and then continued that information over many, many generations. And that seems quite unlikely. It's likely that maybe there is a segregating modifier that's present at different frequencies in these three populations, and that's why all three groups are differentiated. And the third signal we think is likely to be driven by archaic ancestry, and this shows how demography can also play a role in differentiating mutational patterns across human populations. With that, some implications to think about. We often consider mutation rate as this fixed parameter in evolution. We use it in all our analysis, we use it in our simulations, we use it for dating evolutionary events. But its mutation rate is really not one single parameter. It depends upon the context in which the mutations are occurring, and it seems to be impacted by many different factors like demography, environmental factors, mutation modifiers, and also overall evolution of the DNA repair and damage rates. And these have non-negligible effects, despite what most of us consider in our models, in shaping the mutation landscape in humans, but also the same processes apply to other species. And this composite nature of mutation rate and spectrum really makes it challenging to try to use one fixed mutation rate as a molecular clock. And we should all be thinking about how we can try to incorporate these systematic effects in building better tools for dating evolutionary events. With that, I would like to thank my collaborators, in particular Ziwei Gao and Yulin Zhang, and my lab for helpful discussions and funding sources for supporting our work. And thank you very much for listening, and I'm happy to take any questions.
Thanks. If you got questions, you can put them in chat or, raise your hand or something, or just say something.
Priya, great talk. I have a question about, I don't know much about the literature. I was wondering, are all those studies on like generation or age and of the parent and mutation rate, are they all done in Europeans or?
Yeah, majority of them are done in Europeans. That's one of the reasons why we only compare the spectrum in Europeans with the pedigree data. There is one study that expands the pedigree surveys to more diverse populations, looking at Amish populations and Latinos and African Americans, but it's still a pretty small study. And so it's very hard to very precisely characterize these patterns in that case.
I see. And even, like between different studies or data sets, are there big differences or do those seem stable?
There are many big differences and technical issues related to these results that I showed. In fact, even across studies based on the same samples, there are differences. There is, in our analysis, we first did this analysis with using Decode's 2017 data set. And then we looked at Decode's 2019 data set. The 2017 data set has 1,500 pedigrees. The 2019 has 2,500 pedigrees with largely overlapping samples. But in between, Decode changed a filter. And so the two published studies on the same data are inconsistent with each other. So while de novo mutation analysis is really exciting and provides a really valuable piece of information in understanding mutation rate and processes, it's been a bit challenging it's a needle in a haystack problem, and it's quite sensitive to technical factors.
Great, thank you.
Just to follow up on that question, I wonder how much the twofold difference that you observe between pedigree studies and phylogenetic studies is the result of false negatives.
Yeah, I think it's still not fully clear how much of it is related to false negatives. There's definitely an effect of that. However, people have also estimated similar rates when you consider multi-generational pedigrees, or if you consider exome sequencing data. And so slowly those estimates are converging. I think the estimate from pedigrees is probably not 1.2 times 10 to the minus 8. The more recent studies suggest a 20% higher number, but definitely not a twofold higher number. I can stop sharing my screen so it's more a conversation.
I have a question. Have you looked at how much this stuff varies along the genome?
We haven't looked at how much the stuff varies along the genome except for controlling genome-wide patterns. So what we have done is we've tried to ensure that the signal is not driven by, say, recombination rate variation. And so we've looked at different bins of recombination. We have tried to ensure it's not driven by background selection by looking at putatively neutral regions. So looking at these background selection scores, the B scores, looking at it in different bins or looking at higher values of B score, which is neutral. Aside from that, I think we don't really have the power to really look at regional differences across populations. And so most of our regional analysis has been done to sort of control for factors.
And what about the X? Was the X in those plots?
No, we didn't look at X. I feel like since my PhD, I'm always afraid to look at X versus autosome comparisons. And so I tend to, even in this analysis, we had to deal with a lot of technical issues. And in fact, it's only with the high coverage 1,000 genomes data that we started getting confident signals that or signals that we can be confident about. And so far haven't looked at X, but I'm looking for a brave student or postdoc to tackle that. Somebody braver than me.
I'll ask a question. If we were to ambitiously try to update our population genetic simulation programs to reflect this mutation rate variation, evolution and mutation rate stuff, what's the first thing we should change?
I feel like Peter can probably answer this question better because I gave a similar talk at a PopSem meeting several years ago to try to convince people to update the population simulators to account for different types of mutations. I think Kelly Harris was there and gave a similar talk, and there was, I think the current versions of MS Prime account for the differences. So you can simulate different types of mutations. I don't know if you can simulate changes temporally, but I think you could possibly trick the system by generating data for some fast evolving and some slow evolving mutation types together.
Very cool.
Thanks, Peter.
I have another question.
Yeah.
So I feel like I feel kind of disappointed that GC bias gene conversion doesn't pop up as being a more important thing, but is it really just because it's balanced out by the methylation thing?
So I think GC bias gene conversion does pop out if you just compare mutation spectrum across populations. So the RELATE study shows that GC bias gene conversion has a big impact in impacting the rates across populations. In our case, we try to control for it. I don't know if we are perfectly controlling for the effects because while we think that T2C to T2G should cancel out or B should cancel out because they're affected by the same bias, it may not be. In some regions, at least, it could differ. And so we are currently actually looking at this specifically by accounting for, say, strand specificity to see if there are any differences in the patterns. Related to GC bias gene conversion, more generally canceling out with methylation, it's actually quite an interesting question, and something that I've been thinking about a lot lately is the base composition across different species with over very large timescales in evolution actually appears to be maintained in a very narrow range. So GC proportion is between like 38 to 50% in most species. And as far as I know, the only canceling factor is to bias gene conversion is methylation. And so it's actually quite an interesting question to figure out how the two are maintained in such a fine balance. And what are some of the mechanisms that allow it to be maintained this way?
So, one question that follows up on this is that, I was kind of surprised as Peter was that bias gene conversion didn't kind of rear its head more strongly. And so as I was sitting here staring at your plots, one of the things that occurred to me is that actually the signature of a million years ago, I should preface this by saying I'm behind the times, but a million years ago when I looked at this in humans, the major signature of bias gene conversion came from the site frequency spectrum difference between classes of mutations. Because you're conditioning on allele age, there is a non-perfect but very strong correlation between allele frequency and age.
Yeah.
So I was really curious in looking at, I was going to save some of this for when I talk to you one-on-one, but I was really curious to look at those older class mutations, those oldest class mutations, because those should be the ones that are most different. That is, high-frequency derived mutations between the A and T to G and C versus G and C to A and T is the most different class in humans. We have like this tail that looks like positive selection. You basically have this like high-frequency tail.
Yeah, so I've also seen this sort of uptick in the 1,000 genomes data also. And until recently, I was convinced that that was because of miscalling the ancestral allele. Now, after having corrected for that effect, we still see differences across populations at certain types. What we have done to try to ensure that, say, uncertainties and relates age calling and other factors doesn't matter is just looked at these effects in the mutation frequencies, which, as you say, is a proxy, probably not a perfect proxy. We still see the patterns. We have tried to zoom into these and try to look at other regions where there are clustered C to G mutations or other types of mutations. And if they are enriched on archaic segments, as you would expect if it was due to gene flow. And we are still trying to process that data. We still don't have results. It does look like they are clustered across the genome rather than randomly being distributed. But we are still not sure if we are controlling for recombination and other factors fully. So yeah, that's quite interesting. I think a priori, we are controlling for biased gene conversion. So unless you think the bias for T to C versus T to G types is not the same, then it's possible biased gene conversion could affect these. But wouldn't you think that the biased gene conversion across human populations should differ more at recent time scales? Because that's when the population's differentiated. So if we consider an argument that any differs across these populations, the selection differs across the populations, then you would expect that that should be after out of Africa migration, or at least at more recent time scales. Maybe there's uncertainty in exactly when out of Africa migration happened.
Yeah, I think there are two parts. I think you are correct. I think you are accounting for biased gene conversion to a first degree approximation anyways, very well.
Yeah.
The second bit about when I would expect a difference, I don't know. I have very little intuition. One of the things that I would love to see is like a split, one thing that would be convincing, and Peter was asking for this and Murillo as well, like what happens if you look at different components of the genome? So, if you took the top decile versus the bottom decile in recombination.
Yeah, so we see exactly the same pattern as genome. So that that's
cool, right? Yeah.
Yeah, we have looked at recombination rate bins, looked at the top and the bottom decile matching for the proportion of the genome, and the effects are exactly the same. Yeah, another interesting signal, which I'm not still fully sure about, but I can share, is that we have also tried to partition the signal by focusing on sites where Neanderthal is derived, but Denisova is ancestral, or Neanderthal is ancestral and Denisova is derived. And when we call these ND01 or ND10 sites, when we remove either, we don't, the signal is still present. But if we remove sites where both Neanderthal and Denisovans have the derived allele, the signal goes away. And it's actually a smaller subset of markers. And so the signal must be more enriched at those sites, that even by removing a smaller subset of markers, we find that the signal goes away. So one thing that we can tell is it's not related to Neanderthal or Denisovan. It's related to the common ancestor of all three populations, modern humans, Neanderthals, and Denisovans. We haven't ruled out ILS actually, except for the Shankararam work, where they did simulations to show that the site frequency spectrum is explained more by gene flow, and ILS does not explain it. So we are just drawing on their model as this being ghost gene flow, but in our analysis, we haven't actually ruled it out. Again, we are looking at the spatial distribution to see if we can find blocks. But it's also possible that the gene flow happened so long ago that we might not see blocks remaining. So.
I have one more quick question, or
Yeah.
maybe it's quick. So do you think that these genetic modifiers that seem to be changing in frequency in different parts of the world are, is it selection or drift?
Yeah, I'm more of a neutralist, so I would say it's drift, but I would be happy to be wrong. So I think if we can prove it's selection, that would be interesting. A priori, I would believe the null model, or I would assume that it's drift.
Can't you do some back of the envelope calculations to figure out, like, you know, given drift could only have moved the allele frequencies by so much, and so then how big should the difference between the alleles have to be to cause the differences that you see?
Yeah, I actually, so Guy Sala's lab has recently published a theory paper on really characterizing this for the TCC signal. I want to say that they find that this cannot be explained by drift. But they do beautiful theoretical work to try to characterize the effects and have a clear model accounting for the population history to look at the patterns. We haven't applied it to our data, but we can to our ratios also.
Are there any other questions? Okay, so let's thank Priya again. And anybody who wants to can stay on the Symlink or come back in 5 or 10 minutes.
Thank you.
Thanks, Priya.
Thanks a
Thanks,
lot.
Priya.
Talk to you in a little bit.
Yeah, see you in a little bit. Thanks. I can stay on.